Hello, I'm Rie Takeuchi. I'm fourth year student at Ritsumeikan University. Our study focused on bile micelle binding of zwitterionic drugs. First, I'm going to talk about the background of our research. Gastrointestinal physiological conditions change after food intake. In particular, bile micelle concentration is increased. Bile micelles are composed of negatively charged lipids. The free fraction of weak base drug is reduced by binding to bile micelles, resulting in clinically negative food effects. However, the interaction of bile micelles with zwitterionic drug has not been clarified. In this study, we investigate the effect of bile micelles on the free fraction value of zwitterionic drugs. Next, I'm going to explain the drug properties. Fexofenadine and bilastine were used as model drugs. The pKa of fexofenadine is 4.0 and 9.0. The pKa of bilastine is 4.1 and 9.4. In clinical trials, the AUC of fexofenadine and bilastine were reduced by about 24% and 30% respectively after food intake. Next, I'm going to explain the method. We used three types of bile micelles, which is taurocholic acid, oxbile, and hock bile. And we used different concentration of them. Table 1 shows bile micelle composition in each test solution. We dissolved 500 micromolar fexofenadine and 40 micromolar bilastine in each test solution. Then, we measured free fraction value of fexofenadine and bilastine under the condition of pH 6.5 by dynamic dialysis. We used this dynamic dialysis device separated by cellulose membrane. It prevents permeation of substances with molecular weight greater than 3,500. It can evaluate the passive diffusion of drugs. Each test solution of 1.5 ml was added to the donor site, and blank passive of 1.5 ml was added to the acceptor site and incubated at 37 degrees Celsius for 1 hour. Then, we measured the drug concentration on the acceptor site and calculated flux. Let's move on to the results. These upside data show fexofenadine, and these downside data show bilastine. The vertical axis shows flux. In the case of fexofenadine, each phase shift decreased the free fraction value, and they were close to clinical data, and each twofold concentrated phase shift decreased more than phase shift. In the case of bilastine, each phase shift decreased the free fraction value, but they were less than clinical data, and each twofold concentrated phase shift decreased more than phase shift. Based on the above result, we can say that bile micelle binding can reduce the oral absorption of fexofenadine and bilastine after food intake. Moreover, the free fraction value of fexofenadine and bilastine were dependent on the concentration and type of bile micelles. In conclusion, the free fraction value of the zwitterionic drug was significantly reduced by bile micelle binding. These results are important for evaluating the negative food effect for the oral absorption of zwitterionic drugs. Thank you for listening.